Hello. Thank you for watching today's presentation on choosing the best mouse models for your NASH research. My name is Danielle Fontaine. I'm delighted to share this educational information with you to support your efforts to achieve research excellence. If you have any questions about the content of this presentation, please contact us using the information on the final slide of this presentation. Before getting started with today's presentation, I wanted to share with you some information about the Jackson Laboratory. As an NIH-funded nonprofit organization, our mission is to discover precise genomic solutions for disease and empower the global biomedical community in our shared quest to improve human health. And we do this in a couple different ways. The first way is that we perform our own biomedical research. We have our own research staff with their own private funding, such as NIH grants, that investigate the genetics and biology of human disease. Second, we provide scientific services and resources to investigators around the world. In addition to our clinical and research services that some of you may already be familiar with, we also have some freely available resources I'll touch on in more detail shortly. And finally, here at the Jackson Laboratory, we're very committed to education outreach. In addition to webinars such as this one today, we also sponsor traditional conferences and hands-on workshops, as well as educate students of all ages through courses, internships, and other programs varying from high school internships to postdoctoral assistantships. The Jackson Laboratory is the world's largest repository for mouse models of disease. We are NIH funded to import and cryopreserve as many mouse strains as possible to ensure their availability for all time. Among the more than 11,000 strains we distribute from both our Bar Harbor and Sacramento facilities, over 1,500 strains are maintained as live colonies at any given time. Jack's mice are also the best characterized and well-referenced mice in published research, and much of this can be attributed to the rigorous animal health standards and the unsurpassed genetic quality of the mice here at the Jackson Laboratory. What many investigators may not realize is that the use of our well-characterized and heavily referenced common inbred strains such as the C57 Black 6J or the Bulb CJ supports the development, collection, and distribution of specialty mutant mouse models, as well as provides funding for some of our freely available resources. So some of the freely available resources we have are listed here, and I'll start quickly with the JAX Mice database. For those of you who have looked for available mice on our website before, it might have looked something like this for our search. We have data sheets for every repository strain that we have, and while your initial goal may have been to see if the colony we have is live or the price of the mice, if you spend a little more time on the data sheet, you'll see we have tons of other useful information, such as the genotyping protocols we use here at JAX, phenotype information associated with the strain, development of the strain and genetics associated with it, and even a list of helpful references that utilize that strain. The Mouse Genome Informatics database is a great resource that includes information not only about mice from JAX, but mice from other repositories and investigators. We like to think of this database as our one-stop shop for all things mouse genetics. It's a useful way to search for strains that carry a particular allele, and if they're available through any public repository, JAX or otherwise, you can link out to those resources on the allele details page. The Mouse Phenome Database is a collaborative collection of baseline phenotypic data for some of the more popularly used strains. We have others such as the Mouse Tumor Biology Database and the JAX Clinical Knowledge Base that I won't discuss today, but again, all of these are free online resources available to help you with your research needs. In today's webinar, we will compare the translational phenotypes between a variety of NASH models that have been historically used. We will go through some of the limitations of using mice as models for this disease and discuss some of the new and emerging models that are becoming available. I'd like to start today's webinar with a quick overview of why this has become such a hot topic lately. Liver disease is a growing issue both in the US and around the world and has many possible stages. A healthy liver can undergo fibrosis, which is the main topic we will discuss today. Fibrosis is important to understand because if left untreated, it can progress into cirrhosis and even hepatocellular carcinoma. Non-alcoholic fatty liver disease is the accumulation of fat in the liver and is one of the most common liver diseases in the U.S. Current estimates suggest that about 35% of the U.S. population has non-alcoholic fatty liver disease. The progression from fatty liver to NASH is about 10 to 20% of patients with fatty liver. However, most people who have this condition are unaware that they're at risk for NASH development. Often, patients are unaware they have non-alcoholic fatty liver disease, as there are no overt symptoms, and it's only discovered during routine blood work that would show elevated levels of a range of metabolic markers. If a doctor deems it necessary, a liver biopsy would be performed to determine a NASH diagnosis, or a non-alcoholic hepatosteatosis diagnosis. Non-alcoholic fatty liver disease is the most common cause of chronic liver disease, and it's closely associated with many other metabolic dysfunctions. Obesity is one of the most common risk factors for non-alcoholic fatty liver disease, and as more patients present with higher levels of visceral adiposity, which is metabolically active, the risk of liver disease increases dramatically. Non-alcoholic fatty liver disease and NASH themselves are also risk factors for obesity, kidney and cardiovascular disease, and type 2 diabetes. Interestingly, about 3/4 of type 2 diabetes patients have non-alcoholic fatty liver disease. But it's not well understood whether most people develop type 2 diabetes and subsequently increase their risk of non-alcoholic fatty liver disease, or if patients have fatty liver, which then leads to type 2 diabetes. Moreover, a patient diagnosed with type 2 diabetes is at an increased risk of progression from non-alcoholic fatty liver to NASH compared to a healthy patient. Both diseases take place in the setting of obesity and systemic long-term inflammation that results in a large range of metabolic diseases. So what does it take to cross from non-alcoholic fatty liver disease to development of NASH? This is still up for debate, but the current hypothesis is that it takes at least 2 different assaults or multiple hits to overcome the fatty liver disease threshold. The first hit is usually prehepatic, and this includes obesity, insulin resistance, and possibly microbiome alterations. These are often not enough to generate fibrosis in the liver itself, which seems to require a second hit that's intrahepatic. These intrahepatic mechanisms are a popular area of study and include possibilities such as altered lipid oxidation pathways, abnormalities in lipid droplet formation, and particular attention has been given to researching lipoprotein export, such as very low-density lipoproteins. This multi-hit hypothesis shouldn't come as much of a surprise. The liver is a very robust organ that is known for its ability to regenerate. It takes multiple assaults for the liver to cross the threshold from fatty liver disease to NASH. It's important to realize that there is no single characteristic that provides a NASH diagnosis. It's a compilation of many different features that will altogether inform NASH. NASH is characterized histologically, with pathologists looking for several hallmark features such as hepatocellular ballooning, steatosis, increased fibrosis, all of which are shown here on this slide. Fibrosis is generally the most concerning feature, and there is a risk for progression of the tissue to hepatocellular carcinoma and liver failure. In today's presentation, we'll be seeing a lot of histopathology, so I wanted to orient you to some of the common stains and what they can tell us about the liver tissue. First, shown here is H&E staining. H&E staining is a common tool to show inflammation, Mallory-Denk bodies, and ballooning, as you can see by the arrows in A and B. Depending on the severity of these features, a pathologist will assign a score based upon the sample. Serious red staining, not shown here, can be used as a marker for collagen, which is a marker of fibrosis. Altogether, a pathologist will score the liver sample for these traits and will assign a number based upon severity. Using this type of scoring system, for mice, a cumulative score of over 5 results in a NASH diagnosis for mouse models. This is a bit different than the human scoring systems, so you may see some different scores in the published literature. It's important to keep in mind that while mice may be able to model some phenotypes of the disease, it may not be a perfect model. NASH is a particularly difficult disease to model in mice, and though there have been several SNP variants associated with human NASH patients, mice engineered to have these same SNPs did not have the same NASH phenotype. Also, in human patients, NASH develops over a long period of time and is associated with many other comorbidities, such as systemic inflammation, that can have other effects on the body. Most mouse models will rapidly develop certain NASH characteristics without these comorbidities, and these faster models also don't recapitulate the long-term systemic progression seen in human patients. It will likely take more than one mouse model of NASH to develop therapies in order to encompass multiple characteristics of the disease. Here is just one of the many tables you can find when looking through the published literature for mouse models of fatty liver disease. I wanted to bring a few things to your attention. There are many mouse models that have been published upon that will provide a phenotype involved in fatty liver disease: obesity, insulin resistance, steatosis, and fibrosis. As you can see from the table, dietary models, genetic models, and combinations of both can be used. Some of these models, such as a high-fat diet, might have a yes with all of the phenotypes, and this brings me to my second point. Tables like this may be a good place to start when looking for a model of nonalcoholic fatty liver or NASH, but it's incredibly important to go to the primary literature and look at all of these data. A yes or no isn't very detailed. The model might have steatosis, but how severe is it? Even a mild scoring on pathology will still have a yes listed on this table. So again, we always want to make sure we look at the primary publications associated with these models to obtain the best understanding of the phenotypes that are seen. JACS maintains some of the most published upon models of nonalcoholic fatty liver disease and NASH in our repository, including the new MS-NASH mouse, formerly known as the FATSO model. The 4 models I will highlight today are this MS-NASH model, the OB and DB mutant mouse models, and the C57 Black 6J. In the upcoming slides, I will discuss how these mice have been characterized in the literature and some pros and cons to each as a model for liver disease. I want to take just a minute to go through a few of the common diets we'll be discussing. Many different diets have been generated to help promote liver disease, and one of the key points I'd like to make on this slide is that it is critically important to read through the method sections of any papers you might read to determine the actual diet that was used. As you can see here, a high-fat diet, known as a Western diet, can actually range anywhere from 40 to 70% in fat content. The amylin liver diet is known to have high fat, high fructose, and cholesterol. But again, it's important to check on the specific publications to see the makeup of this diet. I do want to point out that there are now many variations of this amylin diet, as this diet is difficult to reproduce due to the FDA ban on trans fats. 2 other diets I want to mention are specific nutrient-deficient diets, both the methionine and choline deficient diet, as well as the choline deficient diet, which has reduced methionine levels but is not deficient. As you can see again, the actual values of macronutrients can range in each diet, which is why it's important to both look at the references to determine what was used, and when you go to publish your study, always include the exact diet used in your research. As the C57 Black 6J mouse is a commonly used mouse model, I'm going to start with this strain. This study investigated placing mice on a high-fat diet to determine the metabolic effect on these mice. One interesting thing I want to point out is this group actually built in a pair feeding system to their experimental setup in order to make sure that both groups, both the experimental and the control, were eating the same amount of calories every day. One group of mice was fed a high-fat diet, while the other was fed a chow diet, but the same amount of overall calories. What the investigators found was that a high-fat diet increased liver triglycerides, which is a sign of liver steatosis. As these mice were fed the same amount of calories, and as you can see by their weight graphs, these mice were not obese, this liver phenotype was caused by the nutrient makeup of the diet and not just an increase in caloric consumption. They performed histopathology on the livers of these mice and were able to show that there is an increase in steatosis, as well as ballooned hepatocytes, shown here with the solid arrows, and Mallory bodies, with the dashed arrows, and the high-fat diet group compared to the control. This models non-alcoholic fatty liver disease really well. However, there is no increase in steatosis or serum ALT levels. So while these mice have fatty liver disease, being on this high-fat diet for 16 weeks does not result in fibrosis, which is one of the hallmarks of NASH. I'm going to move to a study that looks at later time points, but please note that this is the only study I will discuss that uses this pair feeding paradigm. Again, it's really important to look through the method section of any study that you're looking at to determine what the feeding protocol was for their mice, because it may make a large difference. So a different group of investigators took high-fat diet C57 black 6J mice and maintained them on this diet for quite a bit longer, up to 52 weeks. What you can see here is that by the 24-week time point, hepatic steatosis is clear, and by 40 weeks on diet, there's a significant increase in inflammation. Hepatic triglycerides, again a sign of steatosis, was clear at all 3 time points. However, it's not until the 40-week mark that these mice cross the threshold for a NASH diagnosis, as indicated by their non-alcoholic fatty liver disease activity score, or NAS, shown here. As I mentioned earlier, the combined score of criteria needed to exceed 5 before this is considered a NASH diagnosis. 24 weeks on diet doesn't provide this, but by 40 weeks, it's there. This is a nice step showing that these mice, if left on diet long enough, might recapitulate more characteristics of a NASH phenotype, and also models the slow progression seen in human patients. However, many scientists are frustrated that this is such a long process and wanted to look for ways to induce this phenotype more quickly. One method to induce rapid liver damage and fibrosis is by feeding mice a methionine and choline deficient diet. Methionine and choline are important for hepatic mitochondrial beta oxidation and production of very low density lipoproteins. By exposing mice to this diet, liver metabolism is disrupted, and these mice very quickly develop fatty liver disease and fibrosis. From the histology, we can see the lipid droplets in the methionine choline deficient diet mice compared to both a control diet as well as a Western diet. MCD diet promotes fat storage in the liver. When we look at liver enzymes such as ALT, AST, and ALP, we can see that while these markers for liver damage are all increased in the Western diet cohort, they're much more robustly increased in the MCD group. I do want to point out this is being a great example of being cautious of tables you might see discussing mouse models of NASH or non-alcoholic fatty liver disease. Technically, both the Western diet and methionine choline deficient diet have these increased markers, but one is clearly more severe than the other. But just looking at a binary table, they might both just say yes under liver damage. Another reason going through the primary literature will be helpful in picking the best model for your research. One thing not shown on this slide, but I will show you in just a minute, is that these mice are also rapidly losing weight. As you can see, this is also reflected in their liver weight. Often the liver is storing fat and perhaps becoming fibrotic, which can both increase liver weight, seen here in the Western diet fed mice. However, the methionine choline deficient diet fed mice are actually losing liver tissue mass on top of being damaged. One of the reasons this diet is very popular to use in the literature is due to the similarities between mice fed methionine choline deficient diets and human NASH patients in regard to expression of antioxidant enzymes. Oxidative stress is thought to have a large role in the progression of human NASH, and this model mimics the decrease in antioxidant enzymes observed in the human population. You can see this decrease in mRNA and multiple antioxidant enzymes. And when we look at histology, using 4-HNE, which is an aldehyde product of lipid peroxidation, commonly used as a marker of oxidative damage and lipid peroxidation, we can see this is significantly increased in both the methionine choline deficient and Western diet groups. Overall, this might be a useful model if you're looking at the possible mechanistic pathways that might lead to liver fibrosis. As I mentioned earlier, you can see that these mice have a rapid decrease in body weight once given this diet. They also have decreases in leptin levels, which is not surprising as leptin is made by adipocytes. But they also have severely decreased blood glucose levels. This is actually the opposite of what's observed in the human population. the majority of human NASH patients are obese and hyperglycemic. So while this might be a good model for certain aspects of the NASH phenotype, this might not be the best model to choose if you're also interested in the effects of other metabolic comorbidities and their relevance to NASH treatments. To get around this weight loss, a diet was generated that's deficient in choline and contains reduced methionine levels, which, as you can see here, when fed to mice does not result in rapid weight loss. These mice still don't become obese. You can see they're smaller than the control group, but they also do not decline the way the methionine and choline deficient diet group did. Not shown here, but the blood glucose also doesn't dramatically drop. These mice never become hyperglycemic, but they also don't have hypoglycemia. They also have increased markers of liver damage, and when we look histologically using a trichrome stain, which stains collagen, a marker of fibrosis, we can see that mice on this diet develop mild fibrosis after 12 weeks on diet. This model is a bit more refined than the methionine choline deficient diet and might be more translationally relevant. One other model I did want to mention due to its increasing popularity is what is often called a STAM mouse. This model uses the C57 Black 6J strain and treats mice at postnatal day 2 with 1 dose of streptozotocin. For those of you who may not be familiar with STZ, it's toxic to pancreatic beta cells and is often used to generate models of hyperglycemia. As a quick summary, when STZ is injected into the mouse, it targets the pancreatic beta cell, as it's selectively taken up by GLUT2, but not other glucose transporters. Beta cells have relatively high levels of GLUT2, which results in DNA damage, leading to apoptosis and necrosis of a subset of the beta cell population, leading to a lack of insulin secretion and hyperglycemia. When this is used in combination with a high-fat diet, it can provide a model for liver pathology. When mice are injected at P2 with streptozotocin, we can see that they tend to gain weight less than their untreated controls. However, when we wean these mice onto a high-fat diet, their body weight does increase. Not as much as the group where they received no STZ, but were weaned onto a high-fat diet. The STAM mice, as well as the STZ controls, exhibit hyperglycemia, have increased liver weight and liver triglyceride levels, which are both signs of steatosis. Looking more closely at the livers of these mice, we can see the STAM model shows more severe hepatosteatosis and a higher degree of fibrosis when compared to either the STZ or high-fat diet controls, shown with the H&E and Sirius red staining here. These mice were on the diet for 10 weeks, and compared to some of the other dietary-only interventions, we can see that these mice develop fibrosis much faster than some of the other models we've discussed. One of the poll questions we often ask, in order to get to the take-home point that some diets may have different phenotypes, are which diets might result in obese mice? And the options we give are a high-fat diet, amylin diet, methionine choline deficient diet, or all of the above. And the reason I like to ask this question is to really take home the point that not all liver phenotype diets will cause mice to gain weight. In this instance here, the high-fat diet and the amylin diet will both cause obese mice. However, the methionine and choline deficient diet will actually result in rapid weight loss in your cohort of mice. So as a quick summary of what we've already discussed, when using the C57 Black 6J mouse strain, we can obtain several important non-alcoholic fatty liver disease and NASH phenotypes in a few different ways. On a high-fat diet, steatosis and liver damage is observed, along with obesity and glucose dysfunction. These studies tend to take several months before mice have these characteristics, and there is only at most mild fibrosis. Using more defined diets, such as the methionine and choline deficient diet, rapid fibrosis is observed with markers of oxidative stress that mimic patient pathogenesis. However, these models often do not present with the obesity and hyperglycemia that human patients tend to have. By adding in chemical treatments, such as streptozotocin, models can be generated that give mice with mild fibrosis more quickly than the standard high-fat diets, but still don't recapitulate all of the phenotypes observed in human patients. Certainly, these mice are widely used models for fatty liver disease, but given some of these caveats, a search for other models began. One set of metabolic models that investigators turn to when looking for possible fatty liver phenotypes are the leptin-impaired models. The OB mutation is a point mutation in the leptin gene itself, while the DB mutation is actually a mutation in the leptin receptor. Both mutations disrupt the normal leptin pathway and result in obese and diabetic mice. In historical reports, these mice didn't develop NASH when fed a standard chow, but as we discussed earlier, a second hit might be necessary to develop fibrosis in the liver. A group of investigators challenged the OB mouse model with the amylin diet. This diet, if you remember, contains high fat, high fructose, and added cholesterol. I'll go through the labels with you to avoid confusion. Here they have lean C57 black 6J mice fed with chow, and these are listed as lean chow, or fed the amylin diet, and these are listed as DIO-NASH, D-I-O-NASH. The OB mice were either fed the chow diet, OB chow, or the amylin diet, listed as OB-NASH. You can see that the amylin-fed C57 black 6J mice gained weight in comparison to their lean chow controls. Both sets of OB mice gained significant weight, as is expected with the OB mutation, without a significant difference between the amylin and chow groups. When we look at liver weights, both amylin groups have higher liver weights compared to their respective chow controls. However, the OB mutation fed the amylin diet has significantly higher liver weights compared to all other groups. I do want to point out that the amylin-fed C57 black 6J mice do have an increase in liver weight, but this is really exacerbated by the amylin diet. When we look at liver histology using serious red staining to mark fibrosis, you can see that both the OB mice on chow as well as C57 black 6J mice on the amylin diet have some fibrotic tissue. However, this is clearly a significant increase in the OB model on the amylin diet. The fibrosis stage is scored here. The amylin diet may be providing the second hit that's required to develop liver fibrosis and NASH. In addition to liver fibrosis, both C57 black 6J and OB mice on the amylin diet also exhibit an increase in hepatic triglycerides and total cholesterol. Again, the amylin diet increases both of these parameters, but it's much more robust in that OB mouse model. This model may be useful in recapitulating the metabolic dysfunction observed in human patients, in addition to presenting with liver fibrosis. When this OB model, as well as the DB model, are placed on a methionine and choline deficient diet, which we know is a severe, fast-acting model of liver fibrosis in wild-type C57 black 6J mice, both leptin-impaired models on the C57 black 6J background develop hepatic inflammation. You can see here the control mice are on the top line, with the homozygous DB and OB mice below. The arrows are pointing to areas of increased inflammation, and when we look at markers of liver damage, such as ALT, there is also significantly increased in the mice on the methionine choline deficient diet. It is further increased by the DB and OB mutations, with the most significant levels in the homozygous DB mice. Interestingly, it's been shown that DB mice, but not OB mice, develop more severe fibrosis on the methionine and choline deficient diet. Histology shown here, looking at collagen deposition, shows much greater amounts in the DB model on the MCD diet compared to either the control or the OB mouse on the same diet. Investigators quantified collagen 1 mRNA in these mice and were able to show a significant increase in the DB mutant model. It's important to keep in mind that both of these mice are leptin impaired, but the mechanism by which they are mutated may play a role in phenotype. The leptin DB mutation is in the receptor, which may result in other phenotypic differences. It's known, for instance, that leptin DB mice have decreased wound healing ability. This might be one possible reason these mice more readily develop fibrosis than the OB group. Overall, these mice may be able to model certain aspects of NASH that investigators can attempt to target. However, it is important to note that very few human patients actually present with these specific OB and DB mutations, again, making the translational relevance less clear. So overall, these leptin-impaired models nicely mimic the obesity and hyperglycemia seen in human populations. And on the amylin diet, the OB mice have increased hepatic triglycerides, cholesterol, and fibrosis. On the more severe nutrient-defined diets, both mice develop inflammation and steatosis, with DB mice progressing to mild fibrosis. As these mutations are not commonly observed in human patients, these models might be useful when looking at comorbidities such as obesity, but may be less useful when looking at mechanisms of NASH progression. More recently, the MS-NASH mouse model was generated to help better mimic the human NASH pathogenesis. This model has also been published under the name Fatso mouse. This is a recombinant congenic mouse model generated by selectively breeding C57 Black 6J and AKRJ mice. At every generation, selectively breeding for obesity, insulin resistance, and hyperglycemia. Male MS-NASH mice spontaneously develop metabolic disease on a regular chow diet, and if given a high-fat diet supplemented with fructose in the drinking water, these mice will develop liver phenotypes. As this model has an intact leptin pathway, it may more closely resemble human NASH patients compared to the previous models we've discussed. The MS-NASH model fed the high-fat or Western diet supplemented with fructose is written here as WDF for Western diet and fructose. These mice will develop hepatocellular ballooning, inflammation, and mild fibrosis by 16 weeks on diet. Not shown here, but steatosis occurs within the first 4 weeks of treatment, suggesting rapid liver dysfunction in this mouse model fed this diet. As you can see here, body weight of the Western diet and fructose mice increases dramatically. And while the control diet MS-NASH mice are still obese, this is exacerbated by that Western diet with fructose. Within 4 weeks of mice being on the diet, there's a marked increase in liver damage markers compared to the control shown here with ALT levels. This model is a new, rapid model of liver dysfunction that presents with phenotypes more quickly than some of the traditional NASH models used. This model also mimics many other metabolic phenotypes observed in human NASH patients, such as increased cholesterol, increased liver triglycerides, and liver weight. When pathology is scored, not shown here, the nonalcoholic fatty liver disease activity score, or the NAS, is over the threshold of 5, indicating a NASH-like phenotype. In another experiment, the compound obeticholic acid has been tested on these mice. Obeticholic acid is a leading potential therapy for NASH treatment, and here we can see that mice given this treatment, shown as OCA, do not lose any body weight, but there is a significant decrease in ALT and AST levels, suggesting improved liver function. In addition, OCA treatment decreases total cholesterol and LDLs. Again, both of these are increased in human patients and often lead to adverse metabolic outcomes. There's also a decrease in liver weight and hepatic triglycerides, suggesting some improved liver metabolism and possible decrease in steatosis. When the liver is scored, we can see that while there is a significant decrease in ballooning, there are no other significant changes to inflammation or fibrosis. This makes a critical point. Even though blood chemistries might suggest a decrease in steatosis and severity of disease progression, it is necessary to look directly at the liver for a better understanding of these results. While OCA treatment does seem to reduce certain aspects of disease severity, it is by no means a cure. However, having access to models that recapitulate many of the characteristics of human NASH is an important step to discover possible interventions and treatments. So in summary of the models we've discussed, this table compiles each model, treatment, and some of the phenotypes we see. As I do keep mentioning, it's important to look at each individual data set to determine the severity of the listed phenotype. Here we just have listed any positive phenotypes in green, negative in red, and phenotypes that seem very contradictory in the published literature are shown in yellow. As you can see, there are many different models that can be used depending on the phenotype you're most interested in studying. Based on some of these discordant results that are seen and the general variability often seen in metabolic studies, I did want to briefly mention two things. One, JAX is in the process of performing internal validation on many of the models, diets, and interventions I've discussed today. Once we have our results available, we will be presenting a brand new webinar to share them with you, hopefully in mid-2020. Second, we are all aware that the microbiome can play a large role in many different phenotypes, especially in the metabolic field. While we do not provide microbiome-defined mice at JAX, we have looked among all of our rooms and facilities and are able to say that JAX mice display a more similar microbiome between rooms, health statuses, and facilities. Knowing that consistency is key to obtaining reproducible results, obtaining mice with more similar microbiomes may help reduce variation in your experimental outcomes. I've linked to the blog below that discusses the results that we found looking at the microbiome across many of our different rooms and facilities. Here are some up-and-coming models being developed and tested for possible NASH phenotypes. Many of these are available from JAX. We do have an entire section of our website dedicated to NASH, both traditional models as well as these emerging models. I encourage you to read through some of the published literature associated with these strains to determine if they have any of the characteristics you might be interested in. Overall, the biggest take-home point I have for today's presentation is that there are many different mouse models capable of recapitulating different aspects of the human NASH phenotype, and you may have to use multiple models to answer your research question. I do also want to quickly let you know that, as I've mentioned previously, JAX is in the process of performing internal validation on several of the models we've discussed today. By mid-2020, we hope to share all of that validation data, as well as make services available for studies for mouse models of NASH. Please contact us if you're interested in any additional information about these potential services. JAX does also have many metabolic in vivo capabilities. Feel free to reach out to us if you want to discuss any of the things listed on this slide further or any possible projects that you're interested in. In conclusion, NASH pathogenesis in humans occurs over years to decades and is associated with many metabolic comorbidities. This disease is not able to be fully recapitulated in one mouse model, and therefore multiple models may have to be needed to validate any preclinical therapies. Utilizing multiple models will provide more reliable, translatable results. And with that, I would like to thank you for joining us for this webinar. The Jackson Laboratory provides a wide range of services that may be able to help you plan and perform your studies. From providing mice to genome scanning and model generation services, we are always happy to talk with you about your planned research. Thank you for watching this video. I hope that this presentation has provided valuable information to help you achieve your research goals. If you have any questions about the information shared today, please contact us using the information on this slide. We would be delighted to support you with technical assistance.